Hello, everyone. Thanks for coming. Welcome to CSRC Colloquium for today. It's my really great pleasure to introduce Professor Antoni Luque from Math Department of SDSU and also Viral Information Institute. I say he's from Math Department, but actually he's mathematical biologist, and he's really truly interdisciplinary and maybe probably one of the most popular persons in community, covering from math, biology, physics, chemistry. He's basically everything, and he has got expert to combine all of those things. And like I said, he has established successful mathematical biology research, doing so many stuffs with successful external NSF grants as well as internal grants. And one really good thing about which mathematicians usually dream of this, he has published in really high impact journal, including Nature's and those kind of things. So before I say too many things, I'll just let Tony to start. And one thing, if you have a question, you can also write down in the chat box, or however Tony feels that it's okay to disturb, or you want the question at the end, maybe I'll just let him lead his session. Okay, Tony, it's all yours.
Thank you, Naveen, for the flattering introduction. I think that the talk will go down from here. I probably won't be able to be up to the standards, but it's always a pleasure to present here in the CSRC students, colleagues. My lab is interested in the architecture and the ecology of viruses, and we use computational and mathematical modeling to investigate that, and of course, a lot of collaborations. Our goal is to understand how the architecture of the viral capsid constrains and shapes the functions that viruses can have in the environment. And today here, I will introduce our progress on the mathematical characterization of icosahedral capsids, which are the most abundant in the environment. As a spoiler, I have to say, coronavirus doesn't adopt icosahedral capsids, so it's not going to be a main player today. But if we have time at the end of the talk and anyone is interested in it, we can discuss a little bit how the structure of coronavirus plays a role in the pandemic. So in order to understand the significance of why studying icosahedral capsids, I'll give some foundational ideas on viruses first. And please interrupt me at any time if you have any questions. I have a few points in the talk also that I will stop to catch up. So let me see if, I guess that, okay, now I can control it. Good, so viruses are the most abundant biological entity on the planet, and they need a cell host to replicate. I'm just moving a tool here. And so here we have some virus representatives, and here we have some cells. In this case, they're bacteria, and this would be like a gut. Now, viruses need a host to replicate, and this has some devastating consequences, no? And we're going through a pandemic of a virus, coronavirus. We had other pandemics before. We had like AIDS. But the disease is only the tip of the viral iceberg, and viruses play an enormous role on the planet. They regulate the biogeochemical processes at planetary scales. They control the microbial communities that dominate ecosystems, and they also promote the evolution of the genomes and the functions of the hosts that they infect. So the majority of viruses rely on two major components. These components are the infective genetic material that encodes the information necessary for the virus to infect the host and replicate, and a protective shell that for the most part is a protein shell, and it's called the capsid, and it's built of multiple copies of a single protein or a few proteins that are called capsid proteins. And these are going to be the main actors that we're going to be discussing today. The fact that the virus produces the capsid in this way, it's actually extremely important because the amount of volume that it has is small. So using one single gene and multiply and copy it many times to produce a structure that big, it's much more effective economically from a genetic point of view than encoding the whole surface that would require that capsid, which, by the way, wouldn't fit for the most part in most viruses. So there are about a variety of capsid shapes. Here I'm showing 4 of the most common ones. You have a lot of viruses that are rod-shaped. This is very common in particular in plants. The majority of viruses adopt quasi-spherical capsids, and most of them are icosahedral and in the symmetry that they organize their proteins. And this is happening across the domains of life, viruses that infect organisms across the domains of life. You have elongated capsids as well, a certain fraction of viruses, specifically in plants and microbes, they're more prevalent. And then you have a bunch of viruses that are pleomorphic, that they don't have that well-specific shape. Some of them, like the one that I'm showing here, has conical shapes. And the reason I'm highlighting it is because although it's not that common, it's important for humans because HIV forms a capsid that has this shape. Now, the capsid and the capsid protein and the capsid itself are essential in the life cycle. When a virus infects a cell and starts producing the new particles, one of the elements is producing the capsid protein. And these capsid proteins need to interact with each other to form spontaneously the capsid shell. And the genome, it's packed inside this shell, and some viruses co-assemble the genome and the capsid that is formed. Some viruses pack the genome after assembling the capsid. And once the capsid is released, the capsid is responsible for protecting the genome. And that means that if there are any changes on pH, temperature, salt concentrations, it needs to be robust enough to protect the viral content. And the capsids are also involved in the recognition of the host in many viruses and participate in the way the virus introduces the genome in the host, either by translocating it from outside or disassembling the capsid inside the host. So if we understand how capsid and capsid protein, they're selected within the host and outside the host, and they're essential on the life cycle. Now, this covers the fundamentals that I wanted to cover, and now we're going to get in more detail on icosahedral capsids. So if anyone has any question at this point, we can stop for a second and address it.
So all these different shaped capsids are still all made from the same capsid protein, just their shape that differs?
No. So the viruses have different capsid proteins, and we will discuss that. There are different protein folds that they form, and actually they're very important on determining the shape of the capsid. Does that answer your question, Chris?
Yeah, it does. Thank you.
Okay. So first of all, let's discuss what do we mean with icosahedral capsid. And an icosahedral capsid has its proteins organized following the axis of symmetry of a regular icosahedron. Now, a regular icosahedron is formed with 20 regular triangles, and this structure has 10 axes of symmetry that go through the triangles, and they have threefold symmetry. Threefold, and we can see it here, means that if I divide 360 degrees by 3, I get 120 degrees. So if I cover your eyes and I put the axis here and I rotate the structure 120 degrees and then I uncover your eyes, you wouldn't know if I had rotated the structure or not. If you were to mark some atoms before I started rotating it, and then I would have to rotate it 3 times, and you would have exactly the same coordinates that you started with. In addition to the 3-fold axis, you have the 5-fold axis. You have 12 vertices, and they have 6 5-fold axes going through. So in this case, it's 360 divided by 5. That means 72 degrees. So if I rotated 72 degrees and I cover your eyes, you wouldn't be able to tell if I had rotated it or not. If you mark any proteins or atoms in it, I will have to rotate it 5 times with these 72 degrees to get back to the original positions. And the last one that you have 30 edges on the icosahedron, and they're coordinated by 15 axes of 2-fold symmetry. That means 180 degrees. So if I rotated 180 degrees, you're not going to be able to tell if I rotated it or not. But if you marked anything, I have to do it twice, and then I'm going to be on the same initial position. Now, this is the basics for the icosahedral capsids, and one of the things that I'm going to explain today, I'm going to show, is the progress that we've done at determining the different designs that are compatible with capsids that are icosahedral. In order to understand a little bit more the relevance of this, let me show a few more aspects. First of all, is 60% of the families of viruses form capsids that have icosahedral symmetry. Now, these families are not equally distributed in the environment. If you take a sample of a drop from the sea, or if you sample your mouth or your gut, 80% of the viral particles that you will observe under the microscope will display a capsid that has icosahedral symmetry, okay? So they're actually even more dominant than in terms of the family. Now, the icosahedral structure depends strongly on the protein 3D organization, the protein fold of the capsid protein. Here we have a graph on the right side that what it does is it groups viruses, viral families, based on the capsid protein fold that they use. And there are about 18 different capsid protein folds that have been characterized in the virosphere. And among those, 10 are capable of forming icosahedral capsids, and are the ones that are highlighted here, and include, like we can see here, this one has a lot of viral families, but actually this one that we have here, HK97 fold, is the most abundant in terms of number of particles in the planet. So this means that if we are able to understand how the capsid protein fold relates to the icosahedral capsid, and in particular, we will have a fair amount of the fraction of the virosphere that we will be able to study and understand from a structural point of view. And to get there, however, we need to know what's the possible landscape of icosahedral capsids that viruses could be forming. And that leads us to the next part. And before I go there, I will address any questions because things are going to get a little bit more technical. So if any of these initial parts need to be clarified, it's important.
Yeah, could you just state one more time for clarity what is unknown and what you're trying to discover or learn about?
Yeah, so I will show that exactly in the next slides.
Okay.
So we will see that there is a certain framework, which is exactly what we're going to do now, that describes the possible capsids that are icosahedral, and that we have fallen short on that, and we had to do a new framework for it. That is what is the research that I'm showing here. So let's get exactly on the question. I think that was Chris, or at least that's the video that I was seeing. So the framework that people have used for almost the one that Casper and Cook introduced, and based on the quasi-equivalence principle. And what occurred is in the 50s, there started to be evidence through X-ray that viruses were adopting, many of them, icosahedral symmetry. And it was predicted that they would have 60 proteins, because those are the ones that you need to form a regular icosahedron, like the one that we see here in this panel A. This prediction, however, didn't match the actual reconstruction of the virus a few years later, which actually contained 180 proteins. So then there was a puzzling situation, And Kasper and Cook proposed a very elegant solution to that. And what they did is a very clever mapping of the problem. Instead of solving the problem in 3D, what they showed is that you could study how the proteins could be organized in a 2D lattice and then map it into an icosahedral. And so the way they argued is, if you have proteins interacting in the capsid, if you think of them in a 2D lattice, they would try to optimize the molecular interactions by organizing in an hexagonal lattice, where the proteins will be clustering in groups of 6, forming hexamers. And if you want to form an icosahedron, then you need to introduce 12 pentagonal disclinations that are going to help you get the curvature that you need to close the structure and form the vertices of your capsid. So then they replace the protein that we can see here in dark gray, and those are going to be the regions that you're going to be able to make the vertices. And if you distribute these disclinations across the lattice, you will be able to fold the structure into an icosahedron. That's not the way viruses assemble, but it's a clever way in order to characterize the possible icosahedral structures. Since you have icosahedral symmetry, you only need to determine the distance between one pentagonal disclination, like this one, and the nearest one, like this one or this one. If you use an hexagonal lattice and follow the steps, H for one direction, for example, this direction, and K for this direction through the lattice, you're able to recover where to put the disclination. So in this case, we would do 2 steps in one direction. That will be H equals 2. And that means that you will have a capsid that will be the 2, 0, and that will be the structure that we're seeing on the right. And if you calculate the surface of this structure, in particular the triangle, and you normalize it by the smallest one that you can do, you obtain the T number, which is the index that people have used to classify icosahedral structures. This is given by this Diophantine quadratic equation, H squared plus H times K plus K squared. And this gives you a series of possible icosahedral capsids. A discrete series is an infinite series, but you're not able to do all possible T numbers. So you have T1 for the smallest case, T3 for the next one. When you do 2, 0, you get T4. When you do 2, 1, you get 7, and you have a sequence that keeps growing. One of the most important findings that they did for the field is to relate this T number with the number of major capsid proteins that you would need to build a capsid. And this formula, because for T1 you need 60 proteins and T is proportional to the surface, reads as 60 times T. And T is also the number of different quasi-equivalent positions that you need on the capsid to recover the icosahedral symmetry. These quasi-equivalent positions are in the icosahedral structure. In the hexagonal lattice, they're completely equivalent because they're in 2D. Now, this stoichiometric formula of how many proteins you need on a capsid was crucial to use the theory for more than 60 years, because many of the capsids that people were reconstructing were multiples of 60. So it was very nice to fit it in. However, when you look closely, and this is what we have revealed recently, many of the structures that were getting fit into this framework didn't follow the principles that were building the Kasper and Cook theory. And that's what we're going to see in the following slide. So these are 3 examples. We're going to cover a few outliers to justify how our new framework describes them all. So first of all, you had cases that had the same number of proteins or T number, but they have completely different protein organizations who were not predicted by the Kasper and Cook framework. So this is pariocotavirus. It infects the insects, has 180 capsid proteins. With the stoichiometry of 60 leads to a T3. But when you look closely, the proteins are organized in a triangle, 3 proteins in this case, instead of the hexagons and pentagons or hexamers and pentamers of Kasper and Cook. However, this structure, it can be interpreted as the dual lattice of the hexagonal lattice. So in a certain way, it follows the classic Kasper and Cook. We will discuss a little bit more about the duals in a second. This other example, phage-MS2, infects bacteria, has also 180 proteins, so it was classified as a T3. However, when you look closely, there is nothing here that resembles the original classification that we were describing. The proteins are actually organized in dimers, and these dimers seem to form these rhombs. So that's something that we were not predicting, but since the stoichiometry was the right one, it was fitted as a T3. This case is a little bit more peculiar. It's a virus that infects plants, and it was classified as a pseudo T3, and this is the effort of fitting capsids into the framework that was going beyond. So the reason it was called pseudo is because it had 60 proteins, but the proteins had 3 domains, and these 3 domains actually look like the individual proteins that you had in pariocotovirus. So people were not happy calling it, okay, it's not just one protein, because the proteins seem to have 3 proteins that sometimes are separate, no? So they said, well, let's assume that they have 180 proteins, and then let's call it pseudo T3. Okay, this is one set of outliers. Another set of outliers is when you have minor capsid proteins, because the theory didn't predict that you could have major and smaller proteins building the structure. And these 2 examples follow a similar lattice. This is a phage, is a bacillus phage, it infects bacteria. It was classified as a T9 because it had 150 major capsid proteins, so it fit on the stoichiometry. And because if you take the capsomers, the pentamers and the hexamers, and you follow them, so here's one pentamer, and this is the closest one, you have 3 steps, the formula will give you a T9. But you have a bunch of minor proteins here that actually make the hexamers and pentamers be oriented in a way that is not predicted by the theory either. The same happens with herpes, which is a virus that we are more familiar with, or at least herpes simplex virus 1, this specific structure. The herpes family actually infects vertebrates, or all cellular vertebrates in addition to humans. So this has 960 major capsid proteins. It would fit into a 16. And it has actually 4 steps between the pentamers, but then you have a bunch of minor capsid proteins, and again, you have a reorientation of the lattice. So that didn't fit either, but for a long time, it was classified with this index because it was convenient. The last set of outliers, it's not, there are many more, but we're gonna stop here. It's probably my favorite because this one actually breaks the quasi-equivalent principle. It's completely broken, but they still were classified within the class-per-code framework. Picovirus is a virus that infects animals. It has 120 proteins. If you apply the stoichiometry formula, that gives you a pseudo T2, and it was called pseudo because there is no such thing as a T2 when you look at the equation that determines the T number. And the reason this happened is because when you look closely at the structure, you can see that there are dimers. Here is a protein, this is another protein, and they form this dimer, and here you have another dimer. These dimers don't follow quasi-equivalent symmetry. That means if you put them in the hexagonal lattice, you cannot relate them by the symmetries of the hexagonal lattice. Nevertheless, it was squeezed there. And the same happened with Zika virus, which got very popular in the last 5 years because we had another pandemic going on it. It has 180 proteins, and it was classified as a pseudo T3. But when you look closely, what occurs is it forms these dimers, and then you have 3 dimers here together. They don't follow quasi-equivalent positions. So it's something that actually doesn't really resemble the Casper code. So that's what we proposed recently. And before we get there, I would like to address any questions that anyone may have at this point.
Yeah, those, Tony, those rhombi and the kites, those are Penrose tiles. So is that the sort of the origin of these anomalous structures? Is that it's known that you can tile the plane with these rhomboids or the rhombuses and the kites. So if you superimpose that on the hexagonal, does that explain these structures, or are these not really Penrose tiles?
So the answer is yes. There is a relationship, and that's exactly what we're going to show. At the same time, it's not strictly Penrose tiles in the sense of Those are quasi-periodic, or at least the fivefold Penrose styles. And then, but it's true that the ROMs and the kites can fill the space, and that's something that we're going to see in a second. And it's related to how we solve the problem.
So you have to fudge a little bit to put it onto a finite surface. To put the,
Yeah, well,
yeah, I see.
Okay. Any other?
I just have a quick question. This structures is the possible structure. Does it have to do anything with the infectivity or also infectivity of the virus or mutation of the virus? Is it related to those kind of things or not?
Well, most viruses are very robust, making the same structure. So any changes that you would have related to infectivity would be more variations on the same structure at the molecular level. So you would have the knobs maybe moved a little bit, but you wouldn't, for the most part, change the structure. But, and this is I'm going to show at the end because it's the current research, there is a strong relationship between the structure of the virus and the replication strategy of the viruses, but in a broader range, not on a specific virus and its infectivity.
Tony, I have
Yes.
a question. The viruses that you've talked about so far, or at least the way that you're talking about them, essentially have it as the sphere being formed as if the nucleic acid is already inside. But of course, there's all the viruses, especially the phages, that will assemble the capsid, but then have a vertex that allows for the association with the tail through which the DNA will be put in, or the DNA will be put in through a hole in the capsid, and etc. So there's an asymmetry somewhere. And how does that, are you simply, just for the sake of your talk, essentially ignoring that asymmetry?
Yes. So I am doing, but let's say it quickly on what it is. So these two phages, these two viruses, for example, have they assemble the capsid empty, and then they translocate the DNA inside afterwards. And the way they do it is they actually have a pentamer that is replaced by, Anka knows these very well, but for the rest of the audience that may not be familiar, they replace the pentamers by a set of proteins that are forming a portal, and that portal stays there in that capsid. Actually, you even attach a tail, and that's why they're called tail phages. And so strictly speaking, they're not strictly icosahedral symmetry, but for the sake of the framework, that doesn't change much, because at the end of the day, you're replacing a pentamer by a portal to attach the tail, but the mathematical organization of the rest of the capsid follows the same principle. Okay. So yes, for the sake of simplicity, I didn't point that out. But it would fit regardless on the way we're building the framework. That doesn't have a problem on that. Yeah.
Awesome.
So how do we solve the problem, or how do we provide a solution, a temporary one? Probably because we're always wrong as we move forward in science, but at least we might be less wrong than what we used to be. So the idea behind is that we propose that you actually can do the Casper and Cook construction as far as you have an hexagonal sublattice, not just an hexagonal lattice, but anything that is coordinated by hexagons regularly distributed. So you can have many patterns and lattices that do that. These are the ones that have 6-fold, 3-fold, 2-fold symmetries. And they're very well described. And here are two references that I use a lot. The question is, okay, if we want to do this in a logical way, what would be those that are the most useful to start building the framework for viruses? And the principle that we used in our case was the minimizing the information necessary to code the lattice. And the rationale was, Well, if a virus has a limited amount of space to encode the information in the genome because they need to fit it inside, odds are that the lattices that take the least amount of information to code in the protein might be the most likely to be occurring. Okay, that was the idea behind. So what are the lattices that you can form? So there is a set of lattices that are called the Archimedean lattices that are finite, and that are formed by regular polygons, and they have a unique vertex that once you determine the type of polygons that you have around, the whole lattice is set. And among the Archimedean lattices, you have 4 cases that have a hexagonal sublattice. So these would be the lattices that have the least amount of information required to build the regular lattice. Okay. So those are the ones that we started with, and you say, well, these ones we're going to be able to build a Casper and Cook construction. These are the hexagonal lattice, trihexagonal lattice, snap hexagonal, and rhombic trihexagonal. What was interesting in this case is you can see, and this is similar to one of the cases that we saw before, you have these additional polygons that will allow you to locate minor capsid proteins. At the same time, we argue that it's relevant to focus not only on the Archimedean lattice, but its dual versions that are the Lavelle lattice. And how do you build this dual, and why do they matter for viruses? So the dual lattices, you build them by taking, you see this segment, you take the perpendicular in the midpoint, and then you start doing that for all the segments, and eventually you're gonna get tiles. And these tiles, as Jim was saying before, they're gonna be covering your 2D space. Now, in each of these lattices, you're gonna get a different tile. What is interesting from a viral point of view is that these tiles are unique. So for each lattice, you have just one tile, which means that if you have a major capsid protein that you can, maybe a few of them put together to form this tile, that's all you need to be able to recover all the possible constructions. So it makes it very convenient. The hexagonal forms tiles that are triangles. The trihexagonal form tiles that would be rhombs. The snap hexagonal form tiles that are flores. And rhombic trihexagonal form tiles that are kites, and they come together as a full triangle, which we will see that is relevant for some interpretations. Now, what occurs here is that since we are adding additional polygons in the lattice, and these tiles also have different sizes, the T number gets multiplied by a surface factor. So the hexagonal lattice is just the classical T number, so it gets multiplied by 1. But these little triangles add a factor 4/3 for the trihexagonal, a factor 7/3 for the snap hexagonal, and a factor 4/3 plus square root of 3 divided 2 for the rhombic trihexagonal. So that means that the surfaces are increasing for the same number of hexagonal steps. Let's see one specific example here to understand how this works. So this is one particular set of steps, 2, 1. So I take the pentamer here, I do 2 steps in the hexagonal lattice, and now I do 1 step on the other hexagonal coordinate. And that helps me join 2 consecutive pentamers. This would correspond to a T number equals 7. It would have 60 times 7 major capsid proteins coordinated in hexamers and pentamers, no minor capsid proteins. If you go to the dual lattice, you will have 20 times 7 tiles. These tiles will be formed by 3 major capsid proteins, all right? And this will be, let's say, the classical construction. What do you get with the new one? So you can see that now the factor 7 means that you would be, in terms of surface and volume, a 9.3 approximately as a T number. And you have also additional minor capsid proteins. You have the same major capsid proteins in these 4 because you do the same number of steps and you're coordinating that through pentamers and hexamers. But you have these additional proteins through the polygons that we were discussing. In the dual version of the lattice, however, you have tiles. And these tiles now, they're rhombs. And in this case, you have 30 times T. This T8 is just the fundamental one. In this case, it's 7 all the time. And the number of proteins that you need to cover is 2 times N, and the N means that you actually need 2 proteins to do these ROMs because you need 2-fold symmetry, but you could do multiples of 2. You could do 4 as far as you place them correctly, 6, 8, and you, as far as you put them to have 2-fold symmetry to fit in the ROM, you will be fine, okay? The same for the SNAP, it gets bigger, so you have more minor capsid proteins. When you go to the dual version, the fluores, you need to build them somehow. One protein might be maybe too irregular to make a fluoride, but if you do combinations of proteins, up to 5 proteins at least, you could combine them covering the sides, but intermediate cases would be possible too. The rhombic trihexagonal, the same. You have more minor capsid proteins because of the additional shapes. And then on the tiles, you have 60 times T tiles. These tiles are kites, and they could be formed with one protein. Is hard to think, but if you have the proteins configured properly, as we saw before, for example, 3 or 4, you could form this shape. Now, I think that somebody was asking a question. Oh, I could hear a microphone. All right. Well, so these are a particular example. Now let's see what are some of the consequences of these. So one important consequence that has specific hypotheses is when you lined up now the possible structures, here we see only the regular forms rather than the duals, but it applies on the same way. You have a denser sequence of T numbers, which means that before you have a T1, and that would force you to jump on a surface that was 3 times larger for a T3. But now when we have these other lattices, it means that you could actually, as a virus, maybe could benefit from having these extra lattices to evolve your structure from a T1 to a T3 in gradual steps rather than having to jump severely between these capsids. So that would be one of the hypotheses that would emerge from having these lattices that now can breach classical T numbers. Another hypothesis is, well, we have competing T numbers now. So the surface T4 can be built, for example, from the trihexagonal lattice with a 1-1 coordination steps, or the hexagonal lattice with a 2-0 coordination steps, which means that the virus may have the option to select one shape or the other with the same volume to enclose the genome. And one hypothesis that we're pursuing is that, well, it could be that the capsid protein folds that we were describing might have a preference for some of these lattices over others. And if you count the lattices that we already have described 8 lattices, and some of them could be formed, the tiles could be formed with different type of configurations. So it gives plenty of room for the capsid protein folds to have evolved if they were selecting the lattices. And here I show the lattices with the minimum of information, but the new framework that we're proposing is more flexible than the Casper and Cook, because if we have outliers, we actually have more lattices that we can explore. They just increase the degrees of freedom. So it gives us some room in the future for even adjust as we're learning and as we're getting more structures. So before we go back, any questions? We're going to cover the outliers that we were discussing earlier, and we're going to wrap up with what we're doing now. Okay, so we're going to see back the outliers and see how they fit into this context and what is the type of learning that we're getting out of it. So pariocotavirus was a T3, 180 proteins, and now we have that is an hexagonal dual, everything adds up. What I want to point out is that the capsid protein fold of this virus is a single jelly roll, okay? And that's something that we will compare with tobacco ring spot virus, which was our controversial T3. Now, when you look closely at how this virus is formed, you have these kites that we had mentioned earlier. And it's because you have these 3 domains of the protein that are actually copies of the single jelly roll. So you have a 3 single jelly roll embedded in a single chemical protein. And so in this case, it fits well on the rhombic trihexagonal dual, which is exactly this lattice that we were discussing. And what is interesting is that if you take the domain, since it's the same as the Pariocoto, with this lattice, you would predict that the size of tobacco ring spot virus would be 0.9 the size of the Pariocoto virus. And when you do the measurements, that's exactly what you get. You get 0.91. And another aspect that is interesting is that if you join this rhombic trihexagonal dual and you put the kites together, which in this case are single proteins, when you put them together, they form this nice triangle, like in the case of the hexagonal dual. So what suggests is that maybe the single jelly roll has a preference for the hexagonal dual, and that this, let's say, unusual or unexpected configuration doing 3 replicates of the domain, it's a strategy to use an alternative lattice to increase the capsid from a T1 on the direction to a T3. Okay, and that's the things that we're exploring now. FHMS2 was 180 proteins, was classified as T3, but when we look at closely now, we see that it fits into the trihexagonal dual with ROMs forming dimers in this case, which has 1-1 step, so it will be a surface 4. And in this case, the fold is different. It's a beta sheet unique protein fold. And for the picornavirus virus and the Zika virus, what occurs is that it also forms these ROMs, but it forms the ROMs with multiples of 2. So in the picornavirus virus, you have pairs of dimers. So you have a dimer here and another dimer, so 4 proteins. And that's why they were non-quasiequivalent on the hexagonal lattice, because they actually were filling this ROM, that the ROM is the one that has the quasi-equivalence, but not the individual proteins that form the ROM. And now it would be a trihexagonal dual with a 4th pair and this specific tile. And the Zika virus will have exactly the same lattice, the same structure, but the ROM is formed with a multiple of 3 times 2 dimers, so 6 proteins. No? You could expect that you could form these with 8 proteins as well, and 10, and those will be predictions that we would have. By the way, the folds of these proteins are different, and that's probably why they can accommodate them in ROMs. So they're kind of using the same lattice with slightly different strategies. For the case of the phage, basilisk, and herpes, we already see now that this falls nicely in the trihexagonal lattice. We have these extra minor capsid proteins that explain the orientation of the capsomers, how many, the stoichiometry that you have. What is interesting is these viruses all form the HK97 fold protein. And it seems that this fold has a prevalence for the trihexagonal lattice, but it's exploring it in ways that we're still not quite sure how to predict. So here are 2 cases that we're still scratching ourselves on how to approach it. But lambda phage, which is one of the most classical viruses that we have learned molecular biology from it, organized in an apparent T7, the classic one, the major capsid proteins are organized in that shape, but it has reinforcement proteins that attach on the surface, and they place exactly on the position of the trimers, which means that in this case, you have a part of the major capsid protein underneath this trimer, but it somehow brings back the trihexagonal lattice. And another case is the phage P22, also forms the HK97 fold. And in this case, when you look closely, you realize that it has different densities on the capsid. So although you could fit it into a T7, when you look at the densities, it looks like the different densities align better with a trihexagonal lattice. But we're still not sure how we can classify these up front when these viruses, even in the absence of minor capsid proteins, might be navigating the trihexagonal lattice. So where we're going now, and this is to conclude. So this is what we got recently the NSF funding is our hypothesis is there is a direct relationship between the capsid protein fold, the lattice that the virus is using for the capsid, and the strategy that the virus is using to encapsulate the genome. So for example, we were just talking about this. HK97 fold seems to have a preference for trihexagonal lattice, and it forms double-stranded DNA. The genome is part of what is called the Baltimore class 1. and it packs the DNA after it has assembled the capsid. And it packs the DNA at very high densities, quasi-crystalline densities. There is no other biological entity or organism that packs DNA at such high densities. If our hypothesis is true, and this is our preliminary data, it means that if I go to the Baltimore class 1 and I look for a different genome packing strategy, I should see that the protein fold and the capsid lattice selected would be different. And this, for example, happened in this other group that packs the DNA also in a pre-assembled but kind of co-assembled. There is some new data, but it uses proteins to condense the DNA. It uses internal membranes also to pack the DNA. And in this case, you form double jelly rolls, and they form an hexagonal lattice, a nice hexagonal lattice on the capsid. Another example that we were talking about, the one with Pariocotta ring spot virus. Those were single jelly roll and formed this dual of the trihexagonal lattice. When you look at how they assemble, they form the capsid, they co-assemble with the RNA genome. So they pack everything together. They don't form a pre-assembled capsid. And this RNA is positive single-stranded RNA. So it's part of what it's called the Baltimore class 4 that classifies viruses based on how they encode the genome and they replicate it. So if our hypothesis is true, this means that if I go to another Baltimore class 4, which we have other type of viruses, positive standard single-stranded RNA, but they pack the DNA, the genome, sorry, in a different way, we would expect to have a different fold on the protein and a different lattice, because our rationale is that the lattices and the protein fold have been selected to favor that type of packing strategy. So this is what happens with this MS2 that we were seeing before. In this case, it pre-assembles the capsid, and then it packs the RNA afterwards, and it forms a different fold, and it has a different lattice. So that's where we are now. And the reason this is important for us is because what I said at the beginning is our goal is to understand what's the role of the viral capsids, of the capsid architectures on shaping and constraining the function of viruses in the environment. And one of the challenges is how do we get a good sampling of structures in the environment? These high resolution structures are very hard to obtain. You need to have a host. You need to produce a lot of particles. These particles need to be resistant enough to go through cryo-electron microscopy or crystallization. So our approach is to use this framework that implies that you have a strong relationship between the type of genome, the protein fold, and the capsid structure. And we're showing that there is a strong relationship also on the amount of genome and the size of the capsid. So putting all together, we're able to predict the capsid architecture from a limited amount of genomic information. And sequencing right now is relatively affordable and accessible, and you can sequence microbial communities and viral communities, even if you don't have a clue on what viruses are there and what hosts have these viruses, which means that using this framework, we can start exploring the ecology and evolution of these capsid structures in the environment directly from the environmental sequences that we're obtaining. And that's the direction where we're going. So just to conclude, let me highlight a few people in the lab. Colin Brown is the student that has been developing these new 3D models that I've been showing some figures. Diana Lee and James Molinex have helped, especially on the direction that we're going now on the National Science Foundation grant. And Raiden Torok is my collaborator at York. We both have been working on icosahedral capsids for a while, and we both were aware that we needed a new framework. So we got together to just expand Casper and Cook and have something that we can use moving forward in the future. So thanks for the funding, and of course, institutionally, the CSRC, San Diego State, and of course, the Viral Information Institute, which is where I spend most of the time discussing and trying to convince everybody that capsid is the most important and capsid proteins is what really we should focus on. And just to conclude, we have this tool coming up in Chimera X. Is a very popular molecular software visualization tool, and we're developing a computational bundle to generate these structures. as you can see here in this movie, and it will come out soon so everybody can use it to analyze their viruses and other icosahedral structures. So thank you everybody for your attention, and I will address any other questions that you may have.
Okay, thank you, Tony, for the great talk. It's very exciting and interesting, actually. So if you have any questions, anybody?
I'll ask a question if I'm allowed. I don't know.
Yes.
Sure.
Tony, it'll be easier if you go to the 2 previous slides. Second. I guess, yeah, right there. So you implied that in a way that these different protein folds for the capsid proteins are not easily exchangeable, but I'd like you to talk a little bit more about that. And maybe blend into that how these viral folds fit in with Kunin and Krupovich's sort of viral taxonomy scheme.
So let me see. The first question is that they're not easily exchangeable. You mean that a virus cannot easily change their protein fold to do something else? Is that what you mean?
No. I'm asking, did you imply that if a virus has a single jelly roll fold protein, capsid protein, that virus could not easily exchange that single jelly roll capsid protein with a double jelly roll fold or an HK97 or a beta sheet unique fold, or any of those kinds of interconnections?
Okay,
we
so
know there's some exchanges that genetic exchanges that can happen, right? I'm just wondering what would happen if the virus actually had that kind of an exchange occur.
So yes, I mean, you can always find exceptions and things in between, like viruses that do double jelly roll, like you have single jelly rolls that come together. But what is important to notice is, for example, these single jelly roll have some area. So the idea is, what is constraining so much the fold in the first place? Because we have a high diversity on the sequences.
Yeah.
But when we look at the structural parts, we only have a few folds, no? So that means that they're strongly selected. And one aspect on selecting them is, well, if you're forming icosahedral capsids, you might have to shape in a certain way. At the same time, you need to enclose your genome in an efficient way, which means that your fold may have some areas that might interact. So in this case, these co-assembled cases, they sometimes have packing mechanisms that interact with the fold, which means that if you suddenly duplicate the fold, I mean, that's kind of the mystery question, no? It's like, can they suddenly jump? Do they have some room for maybe exploring gray areas on these lattices so they can do gradual changes?
I mean, I'm assuming that the double jelly roll is actually a duplication of the single jelly roll to some extent?
Yes, but then once you have this duplication, you cannot accommodate your proteins to form the lattice the same way that you would form, let's say, these ring spots. So then suddenly you're organizing your capsid in a different way, and that exposes you from mechanical properties, chemical properties that might vary. And then maybe your strategy of co-assembling with the RNA, it's not the most convenient one in that case, just because the physical constraints that you're suddenly acquiring. The same fold also can be placed in different ways. So you have vertical and horizontal single jelly rolls, and they lead to completely different lattices, and they're associated with completely different genome packing. And related to the question about Kunin and Krupovich megataxonomy that we have now, my way of understanding it is simple. Single jelly roll is the most popular fold among viral families. It's the one that more viral families form. And the idea would be to look at the single jelly roll and associate it to the specific genome strategy. And the working hypothesis that we have is that the lattices are going to be different. So that should help us narrow down that it's not only about the fold, but also about the way you organize the fold in the capsid and the way you're assembling the genome. And we have some examples on that that show nicely that as soon as you assemble the genome or you pack the genome in a different way, your strategy to assembling the protein and the capsid is also different. So that's where we're going at. But yes, it would provide a structural angle to what they have proposed in their mega taxonomy.
Right. Thank you.
Okay, so I just have one quick question about this practical point of view. Like these structures, virus prefer certain structures, and certain structures has to do with the certain replication strategy. And how much it's practiced that, or people have thought that to design a drug to destabilize this structure so that you can stop the replication or not? Actually, have people thought of about that, or you can put something drugs in your computational simulations too?
So that's one of, yeah, that's one of the directions that I'm trying to go on. Now we're gonna develop some molecular simulations, and one of the arguments that we want to use to convince NIH to help us develop that is that if your fold is so constrained to do lattice architecture, it means that suddenly the HK97 fold may have some common pathways on the assembly that, regardless of how different your viruses may be, you could interfere the way you're saying, no? That if you have a certain generalized antiviral strategy for those viruses that use HK97 fold and form a certain types of capsid structures, you would be able to be successful or adapt them successfully. And so that's something that we want to explore. I know colleagues that have been funded either on the experimental or the modeling side, proposing that interfering with the assembly and starting it from a biophysical point of view, it's a good strategy. And so it's something that seems to be appealing. But in our case, we're going to be focusing it on that. The fact that whatever we extract on the full lattice relationship could be generalizable. So that means that if you have a different virus, but it has a similar pathways on that end, maybe the antiviral strategy that you could use could be extended easily. Now, that's at least the premise that we have in mind.
Okay. Yeah. Thank you. Yeah. Yes, please.
That was a great talk, by the way, Tony. My question is, can you say anything about the architectures that you've identified, the shell architectures, and their stabilities or their energetics or their ability to self-assemble?
See, so that's exactly one of the directions that we're pushing now. So my insight on it, I don't have data yet, but we're exploring the Trihexagonal in terms of the energetics because it's clearly competing against the classic ones. So the idea would be to do an analysis of the equilibrium mechanical properties. One lattice, by the way, that doesn't seem to show up too much, and I'm going to show it and relate it to Ree's question. You see this lattice that has these squares here. Based on my experience on the mechanical properties of capsids, you would expect to have a fair amount of mechanical stress if you try to feed a square there. So it's hard to imagine that at least with a well-covered capsid, this lattice would be that useful in this form. It's more useful on the dual. So those would be my expectations on when we do the mechanical analysis. I would say this one probably is going to be mechanically less viable than others. In relation to the assembly, is interesting. When you go on this route on the tiles, you might have more opportunities to do these different cases. You're paying complexity on the shape, but you're competing on same type of structure, let's say on tiles. But when you go on this route, you're paying complexity. I don't know really how they manage to coordinate assembling minor capsid and major capsid proteins. So you definitely better have a good mechanical advantage to do this trihexagonal lattice, because I don't see how on a self-assembly process would outcompete the classic hexagonal one. So that suggests that you probably have some mechanical benefits or host interaction benefits, because on the assembly, I would expect these lattices to be more costly and more have assembly pathways that are a little bit more constrained. Does that answer a little bit on the insights your question, please?
Yeah, that was very helpful.
So in your case, you're working with this, and this is something that maybe we can follow up with RNA viruses, and that will be interesting to see how they are exploring these possibilities.
Okay. Yeah.
Hi, could I ask a question?
Sure.
Yeah.
I'm just curious on, are certain lattices more likely to come out if they happen to have gaps within them to allow for more complex viruses to evolve? Like, say, for example, a gap for a spike protein to fit in?
Yeah, so that's yeah, sorry.
No, continue. I'm done.
So yeah, that's actually one of the challenges that we have is, so the answer is yes, we have observed, and actually we tried to squeeze this in the paper that we published in last year. We have evidence that this lattice is observed in the pro-capsid, in the immature capsid of some viruses. Actually, some of the Flaviviridae, like the Zika virus. The Zika that we saw was the surface, but it has an interior part, and that part seems to follow this strategy. However, you have gaps, no? So we had a hard time really addressing that, and the reason is because the framework, the way it's built right now, and this is one of the let's say the weaknesses of the framework, is it doesn't really follow the assembly process. It's based on the idea that you have a lattice in 2D that you can map into an icosahedron. So the moment you start adding holes, you have a lot of flexibility to add holes, but we don't really know how to constrain how flexible would you be putting those holes. That has a more physical element into it that we would like to explore, but that I don't see that I can provide predictions. I can just say, yes, it's possible to have these lattices with holes, no problem. But I cannot really constrain too much. As soon as you start putting holes, the fact that I'm not covering the lattice, it means that you have a lot of flexibility there. So I can really not predict much. But we have evidence that this lattice, for example, this one here with a different steps, we have observed something similar to that in some viruses. So we're exploring it on the whole side, but it's a more complicated venue.
Can I add on? Are you, it seems as if you're doing everything on one single 2D lattice, but I'm just wondering, are there certain other, can you expand and you look at another lattice and then overlay it on top of the current one that you're working on and maybe see if there's any matching in that sort?
Yeah,
I would imagine that would probably be.
Yes.
Like,
so what we're trying to do now is to go backwards and say, if I have a capsid, well, one of the directions is if I have a capsid, what lattice would I propose without using the framework, just by identifying how the proteins are organized? And the idea is to have a computational approach that helps us explore the complexities, because in terms of mathematical transformations, we could do exactly what you said. Like I could overlay this one here, and I could do it now. It's a little bit late, but the program that we have developed does it. And it does very beautiful patterns, and you have infinite amount of patterns that you can do. The question is, how do you approach those patterns in a way that helps you classify what is more likely to occur and that it gives you prediction power? And that's where we're having the struggle. If we start using all possible combinations, we don't really know where to stop. So start with the minimization of the information, and then from there build up, especially as we get evidence from the capsids. This is like Now that we have these lattices, we look at capsids in a different way, which means that we will see details that before we wouldn't probably tell. So we're probably going to see outliers of these very soon, because we now have these to look at it. And what you say is one of the directions is if we mix these two, gives us some patterns. Could it be that viruses are using that or exploiting that? And I would say probably, but we don't know really how to constrain it right now. Does that answer your question, Daniel?
Yes. And may I ask one more? Just sorry, if that's okay, if anyone doesn't have one. Or I was just curious, are you including texture in any way in your 2D lattices?
We're not yet. And that's one of the directions that has the most problematic part, because and maybe this with these 3D reconstructions is easy to see. You can see that the proteins also, this is another artifact or approximation. You do a 2D lattice into a mapping of a 3D, but the proteins are not really a 2D surface, no? You have a volume on 3D that has some areas that have cavities, holes, some protrusions. And once you add this texture, right now we don't have a way, again, to constrain it. So my hypothesis, the way I want to follow up this and the way is mathematically you have the following problem. You have a single polypeptide that you need to fold in a 3D structure that needs to fit nicely on an icosahedral structure. And there is where you start seeing that you may have some constraints on the surfaces and cavities that you could allow to have, especially if you want molecular stability. But I don't know where to put the constraint right now. So it's something to explore, not right now implied in the surface at all.
Thank you.
Okay, I think yeah, we discussed a lot. It's very exciting. We are looking forward to what you can come up. It's really so many exciting open questions. Okay, thank you very much, all. And usually our tradition is we used to go to Eureka after this for beer. You can still go to your fridge and get very cold beer and enjoy. Okay, thank you very much. Thank you, Tony, for a great talk. Yeah.
Thank you, everybody.
Awesome talk, Tony.
Thank
Thanks, Tony.
Thank you.